The data generated by mass cytometry are complex and need to be visualized in an efficient way and simple manner. Cytofast is a method which highlights immunological patterns within the immune cell population and determines cell subsets which are linked to clinical treatment or experimental group. Cytofast can be applied after any clustering method like FlowSOM or Cytosplore. It will allow you to discover cell subsets which are linked to a clinical treatment or an experimental group. With this method, you will be able to see the immunological overview of your data at a glance in a quantitative manner.
Begin by creating clusters with either Cytosplore or FlowSOM. If using Cytosplore, upload the FCS files by clicking Files and Open FCS files. Then, select a cofactor for hyperbolic arcsine H transformation when prompted, and click on Add Unique Sample Tag as Channel. Select Run HSNE to run an HSNE level of 3 and wait for the map to be generated. On the first HSNE level, check the cells that are positive for CD161 by selecting the CD161 positive cells and right-clicking Zoom into Selection. At the second level, repeat the procedure to reach the third level with only CD161 positive events. Once the last TSNE map is generated, save the clusters defined by Cytosplore by right-clicking on the map and choosing Save Clusters. Choose the directory of the output files as prompted by Cytosplore, and note this location because it will later be used to load FCS files into R. Use a simple characters-only name when renaming the output files, which will make identification and further handling easier.